It gives me great pleasure to welcome to Skeptical our last speaker, Dr. Ioannidis. Dr. Ioannidis is professor of medicine at Stanford University, and among other titles is co-director of METRICS, the Meta Research Innovation Center at Stanford, a center that works to improve the quality of biomedical and other scientific research. He holds an MD and also a DSc from the University of Athens. His training both in medicine and research is reflected in his interests in the quality of scientific research and how it can be improved. His publication with the rather startling title, Why Most Published Research Findings Are False, has been the most accessed article in the history of the Public Library of Science. He is also one of the most cited scientists in the world. I met John a couple of years ago when we were both asked to speak at a Skeptics conference at McGill University, and I thought even then it would be just terrific if we could finally get him to come to Skeptical. And I'm very pleased that we were able to do so this year. Would you please welcome Dr. John Ioannidis.
It's a great pleasure to be here today, and I would like to share some thoughts and also hear probably some questions and suggestions on how we go from here in terms of making research more reproducible. So science has been a success story. It's probably been the best success story of humankind through its history. But we do see lots of results that are not reproducible and not replicated. And that was the theme of a paper that I published 10 years ago that Eugenie referred to. A few months ago, trying to repent, I wrote another paper for PLOS Medicine on how to make more published research true. So my talk will be 50% of the first theme and 50% of the latter. Much of the problem stems from the fact that indeed science is very successful. So we have 15 million different scientists publishing papers in major journals, according to Scopus alone. And this is just 25 million papers out of the 114 million scholarly documents in English language alone across all science and all times. It's an estimate from a paper in PLOS One about a year ago. Since then, probably there's another 5 or 6 million papers that have been published. Each empirical paper can include anywhere from a few up to many millions of results. So we're talking about trillions and quadrillions of analysis and results and claims that can be made. What does that literature look like? If you take PubMed, that includes all the biomedical literature, this is a random sample of PubMed that we looked at, 441 journal articles. Only one study provided a full protocol. None made all the raw data directly available. Replication studies occurred in 4 cases. Only 16 studies had their data included in a subsequent systematic review of the evidence. And only 26 studies mentioned conflicts of interest. So this is the recent biomedical literature at large. What should we look for towards ensuring that more published research is credible? Should we look for significance? Should we look for innovation? Or should we look for rigorous approach to scientific investigation? Significance sounds nice, and it's something that I write in all my grant proposals, that I'm heading for something very significant and important. But the problem is that everybody does that, and everybody publishes that. So across 1.5 million abstracts with p-values in Medline that I looked with David Sevalias, we found 96% of them reporting statistically significant results. This is a paper from Dan Fanelli, who recently joined us at METRICS as a senior scientist. He looked across different scientific fields for the proportion of studies that favor the tested alternative hypotheses. And as you see, some sciences like space science, geoscience, physics have rates that are anywhere from 60-something% to 75%. But medicine, pharmacology, psychiatry, psychology, and economics are 90+%. And according to Dan, this is a a way to say whether a scientific field is soft. So if you have too many statistically significant results, this is not proof that you're more successful. Maybe it's proof that the field is more soft, and therefore there's an inflation problem in the number and proportion of significant results. How about innovation? And obviously in all my grant proposals I also say that I'm innovative, but to be honest, there's hardly any reliable method to predict innovation ahead of time. The vast majority of honest and excellent scientists never produce anything that is highly innovative despite continuous hard work and every good intention. And even when that happens, it takes many years, often decades, to materialize that great promise and to translate itself into something that is useful for humanity. It does happen, but it doesn't happen overnight. And this is an empirical evaluation looking at the most highly cited papers in medicine. The red points is when we have an extremely highly cited paper for an intervention that promises to be very effective in a randomized trial or a big observational study. And you see the prior landmarks: first description of the intervention class, first description of the intervention, first article about human use, first article on specific human use, and so forth. On average, it takes about 25 years to go from the first description that something is being discovered until something does make that difference. And even when it does make that difference, you see that there's also these black boxes about 40% of the time, even for those creme de la creme papers. Other papers that were better done, better studies, better controlled, much larger, refuted these highly cited paper. The typical recipe of research practices varies across different fields. So there are some fields that have highly collaborative work. So high energy physics and particle physics are highly collaborative. Much of astrophysics and collection of data from telescopes is done again with data sharing and with lots of investigators participating in consortia and vast collaborations. But in most fields, the typical recipe is that you have a solo, siloed investigator trying to prove that he or she will come up with something that's significant, innovative, or both. Unavoidably, being alone, the sample size of the studies that I would be able to conduct would be pretty small. To try to get something that looks so significant and so important and so extravagant, I need to cherry pick, usually one or the best among many hypotheses. Much of that will be done post hoc. Most of these fields are looking at very lenient p-value thresholds, like p-value of less than 0.05. There's no registration of ideas or protocols or hypotheses. There's no replication. It's considered as Me Too research. There's no time where we want to move forward. We don't want to repeat what others have done, and no data sharing. So when you get this type of mix, this is what you get. This is an analysis of 1,778 data sets of fMRI studies, a very popular and hot field. And as you can see, the only chance to make lots of discoveries of new signals about fMRI and different phenotypes of things that are happening in the brain is to perform extremely small studies. Once you start performing larger studies, very, very few foci emerge. And this is not just fMRI. About a year and a half ago, we published that paper in Nature Neuroscience, where we looked across very different fields in the neurosciences, and we saw that there's kind of a pervasive power failure. On average, fields of neuroscience investigation were operating with power of about 20%. In some cases, it was as low as 8%. So you expect to have a recipe for disaster and tons of both false positives and false negatives when a field is operating in such low power. Then it's not that we only have one analysis, there's multiple analyses that we can conduct, even on the very same question. So this is a snapshot of about 25 of the 60 studies that have been done trying to identify risk factors for pterygium, which is an eye condition. And you see a line for each study, and you see the 10 risk factors that are considered to be very important, probably everybody agrees. So trying to find new ones we need to adjust for those. But the green color shows what is being adjusted for in each of these studies. There's not a single pair of two studies that have adjusted for the same things. Each one has done something different. Even though there is consensus, or seemingly there is consensus in the field, what is reported is completely different from one study to another. And this is extrapolating the same concept from 60 studies to 1 million studies, but 1 million studies on the same hypothesis. So each one of these is a cloud of 1 million results on the same analysis, looking at the association of things like vitamin D and thyroxine and creatinine and alpha-tocopherol in association with mortality risk. So on the horizontal axis you have p-values, on the horizontal axis you have hazard ratio for death, and on the vertical axis you have a minus log 10 p-value. And how do we get 1 million results for the same analysis from the same data set? Well, how many things affect the risk of dying? Many. So if you consider 17 or 18 of them, and you either adjust for or not adjust for each one of them, and a couple of them have actually 3 categories, if you run the calculation, how many options you have, how many different models you can create about 1 million models. So you can get 1 million different results for the same hypotheses and the same analysis being investigated. And these results can be pretty different, sometimes very substantially so. So if you take a look at the right panel, this is alpha-tocopherol, vitamin E. Vitamin E, for a long while we thought that it was a wonderful thing to get, and it would decrease mortality, it would decrease cardiovascular events, it would decrease cardiovascular deaths, it would decrease cancer, it would just be magnificent. But then we had multiple randomized trials suggesting no benefits, and now we even had a meta-analysis suggesting that at high dose you can even increase mortality. So what happens with alpha-tocopherol if you run these 1 million analysis? 70% of the time, alpha-tocopherol will seem to be protective, reducing the risk of death. And 30% of the time, it will seem to increase the risk of death. We call that pattern the Janus phenomenon, from the Roman god that has 2 faces looking in opposite directions. So depending on what you like to say, you can pick and choose a result that will say vitamin E is great, vitamin E is miserable, vitamin E does nothing. And about 40% of variables in NHANES, the National Household Survey in the US, perform in that fashion, with results being on both directions, just depending on what you adjust for or not adjust for. Empirical studies on fields where replication practices are common suggest that most of the initially claimed statistically significant effects are either false positives or substantially exaggerated. As I said, replication is considered to be me too. We don't want to do lots of that, but when it's very cheap and easy to do, it has been done. And one field where this is very cheap to do is genetic epidemiology. So for a long time, we were cherry-picking hypotheses one at a time, and down the road, the cost went down 10 million-fold. We could just screen the entire genome. So we could go back and see what happened to all these things that we thought we had found when we were cherry-picking one thing at a time. The replication rate was 1.2%. And many scientific fields are still in the cherry-picking phase. So genomics has matured. There's still several studies done in that fashion, but obviously there's lots of much better science being done, which is highly credible. And the replication rates using far more stringent statistical thresholds, large-scale collaboration, large sample sizes, very concrete analysis plans is 99%. But much of that literature that was prehistoric meaning 10 years old, and there's still many studies being done, the replication rate was 1.2%. Who else might want to replicate? Well, very often I've given talks saying that the industry wants to get favorable results for their products, and I say that again and again. But in some types of research, the industry have been champions of replication, in particular when it comes, for example, to preclinical research, where it does make a difference for them to have credible results coming from academic institutions that they can use as a basis for developing new drugs. So many different companies and investigators working in them have looked at a large series of highly cited papers coming from the best academic institutions proposing new drug targets, and the replication rate has been anywhere from 10 to 25%, even involving the original scientists to try to tell them exactly what did you do here and how did you get that result that I cannot get. In the case of Amgen, they could replicate only 6 of 53 landmark studies on oncology drug target projects, and Glenn Begley concluded in his paper a couple of years ago that the failure to win the war on cancer has been blamed on many factors, but recently a new culprit has emerged. Too many basic scientific discoveries are wrong. Along with Glenn, we wrote that paper a few months ago, trying to put together what we know about reproducibility efforts and reproducibility problems in preclinical research. And I'm just showing you that very busy slide to convey the message that it's not just a one-time occurrence. Problems have been seen in microarray data, in stroke animal studies, in general biology, in oncology, in genome sequencing, in vivo mouse studies, neurological studies, mouse models, genomic cell line analysis, microarray data on compliance with standards, Miami standards, commercial antibodies, commercial ELISA, and also in the interface of how results are presented eventually in journals, and that knowledge is being disseminated. And here's a number of fields that have identified problems: neuroscience, pharmacology, genomics, bioinformatics, stem cell biology, oncology in vitro testing, chemistry-led discovery, computational biology, pathology, biomarkers, organizational psychology, and all types of observational research. This is something that was recently announced from a large-scale effort that psychologists are conducting to try to replicate 100 findings from the psychological literature that are very well established, seemingly, and multiple teams tried to look at these findings, repeating the experiments. 39% of those could be replicated, matching the original results. But then there's an extra layer that maybe they were replicated or were not replicated, but they still had some similarity or did not have some similarity. So that was in Nature News. We still have to wait for the final paper. But these issues have been raised in multiple fields that are still working with the recipe that I described to you up front. And this can happen in psychology, it can happen in economics, it can happen in behavioral sciences, in sociology, in political science, and in many, many other disciplines. Many scientific fields are also going through a revolution of big data. We have tons more information to analyze. And this is great news, but as I wrote with Moin Khoury in Science about a year ago, this can also create big noise. And it can create situations where our priors for identifying genuine associations and genuine effects goes down, because we just massively go after things that we had no good reason to go after. So big data, big noise, big error, it's a big challenge to try to get more without really introducing far more noise in the process. And finally, one other dimension is what I call stealth research, which means that currently there's lots of people who are interested in research because they can get money out of that, and they don't want to share their secret or their technology. So for some of the seemingly very innovative technologies, there's nothing to be found in the scientific literature, in the peer-reviewed literature. You may find some patents. Actually, you may find lots of patents, but try to understand what has happened just by reading a patent. It's really an ordeal, and clearly you cannot repeat the experiments. I mean, you can get a sense about roughly what the concept has been, but definitely not have enough to reproduce. So I will spend the second part of my talk trying to discuss solutions that we have to improve research practices in that setting of a reproducibility crisis. There's many solutions, and as these problems have become more evident, which, as I say, is kind of a collateral damage of the success of science, some solutions have started being proposed, some of them have been applied, some have already worked. Others are very speculative, and I think that we need empirical evidence on whether they work or not. I think that we need science about science. We cannot say that we're not going to use the scientific method when we decide how are we going to conduct research. And many of these proposed solutions may actually do harm. So they may be nice ideas, but when we adopt them, who knows what will happen? So we need to find out. The solutions have to tackle the problems that we meet in different steps of the scientific investigation enterprise. So at the level of the design of studies, at the level of conducting studies, at the level of analyzing data, or how results are reported and disseminated. And this is a list of about 12 different types of solutions from my last year's paper. I will run through some of them and highlight some of the promises and caveats that each one of them has. So large-scale collaboration and adoption of replication culture, I think, is clearly a winner. Whenever we know what we are after, I think it's the best thing to do. If we want to discover something that nobody has a clue what it is going to be and how exactly it should be designed, obviously we can't ask everybody to do the same thing. We should allow people to be very creative and search as widely a possible space of investigation. But if we know that all we want to do is 2x2 tables to associate gene variants with, say, a phenotype like smoking behavior, we have 100 teams that are doing this. Let them come together and run a single consortium analysis. And this is what's happening here in that slide. When we did that, we ended up with a sample size of 200,000 people, 100-fold bigger than the typical study that was done in the past. We identified 4 genes that regulate smoking behavior. Before we did that, we thought that we had found 659 genes. So one of the 659 survived, and we found 3 more, and by expanding even further the sample size, we're up now to about a dozen that have very rigorous support. Now, who is going to replicate? Sometimes maybe the same investigators. If you have a big consortium, that could happen within the constellation of the consortium. It could be different investigators of the same school, but then you may have some problem of inbreeding. Some people argue that we need different investigators of competing theories and competing hypotheses, so have your enemies replicate what you did, and then it's end of story. Combinations of the above, or in some cases, replication may be open to the wide public if we have citizen scientists who can access data, replicate data. Depending on the type of analysis that needs to be done, maybe they can do it themselves. What are the credentials for a replicator? I think this is something that we have to be very careful, because maybe not everybody has the credentials to replicate something thoroughly. And in some fields, you really need a lot of expertise to do that, and a lot of background technology and a lot of infrastructure. In other cases, maybe almost any citizen in the world could do it. Registration is another set of solutions. It means that I'm trying to register what I'm going to do so that other people would know that this is what I had in mind and I didn't really deviate in my path. So again, that applies mostly to research that I have some good sense of where I'm heading. In some other cases where I don't know exactly where I'm heading, maybe I should just be honest about it. And maybe what I could do is inform the people about the potential studies that I could do, but I haven't done yet, but I have the power to do, because I have data in my hands, in my computer, in my laptop, and I can start pushing these buttons and start generating millions of chi-squares and destroying the world. So people should know about that I have the capacity of generating so many millions chi-squares by knowing what is my database like. And this is the different levels of registration. Level 0 would mean no registration at all. Entirely blue sky science. I have no clue what I'm going after, but maybe downstream I will come up with the new theory of relativity or something. How would I know? Nobody knows, so nothing to register. Level 1, registration of the data set to know what can be done. Level 2, registration of the protocol. Very explicit. This is the aims, this is the objectives, and primary outcomes, secondary outcomes. Level 3, registration of the analysis plan, exactly how the analysis is going to be done. Level 4, registration of the analysis plan and the raw data. And level 5, open live streaming where you open your computer to everybody to see, and each morning you say, this is the experiments that I'm going to do. Is that a good idea? You wait until noontime to hear what other people think, and then you run the experiments, and by the end of the day, you post the results so that people can see and comment, and next day, same thing. How much of that is happening? So I think the scientific community is increasingly sensitized to the need for transparency. And 4 years ago, we did that analysis where we looked at the 50 most highly cited, highest impact factor journals across all science. And we tried to ask how many of those have policies in place, editorial policies for sharing microarray data in public, or nucleic acid or protein or macromolecular data, sharing materials upon request, sharing protocols upon request, or even making sharing of data, protocols, and materials a condition for publication. Paper will not be published unless you do share all of that. Lots of color, which means that lots of these journals do have policies in place. But the last column has numbers, they're very tiny font, but there's tons of zeros there, which is how many out of 10 papers in each of these journals actually adhere to these policies. So even though the policies were there, people were still reluctant to adopt them in real life. And we are repeating that exercise currently with colleagues from NIH. It seems a little better, but not much better. Sharing is easily said and difficult to do. There's many hurdles to that, and some fields have been more successful than others. Sharing telescope data, can someone make money out of sharing telescope data? I don't know. Probably not so easy. But if it's medical data, one can make money out of selling a drug. And if the data are reanalyzed in a way that they show that the drug doesn't really work, that could be billions of dollars for the sponsor. So along with Peter Doshi and Steve Goodman, we put a list of about 25 challenges on sharing data. Who's going to do that? When it's going to be done? How it's going to be done? What type of data should be available? How much effort do you need to clean, to prepare them for others to use? How much communication you need for others to understand what's happening, what has been done, what each variable means, what are the caveats, what went wrong with the study, when should data be shared, how quickly, who should get credit, how should credit be allocated, should there be incentives, how do you enforce incentives, should there be penalties, should be unrestricted, and so forth. And the Institute of Medicine put together a very nice report a few months ago on expanding on many of these challenges. So people want to move forward, but there's many challenges still on how exactly to do it. Now, let's say that you do have the raw data available from, let's say, randomized clinical trials. So, these trials of drugs that could mean billions of dollars in sales. There are already examples where data have been shared and people have reanalyzed these data with the very same hypothesis, the very same population, the very same comparison, the very same outcome, and published papers with these reanalyses. So along with Shanil Abraham and several other people from my team, we looked and found 37 such occasions where papers had been published saying that we are reanalysis of randomized clinical trials. 35% of the time, the conclusion on whether these interventions should be used or not, patients should be treated or not, or who are the patients to be treated, was different in the reanalysis compared to the original paper. Now, why was that? Maybe one might think that it was some rogue reanalyst, some enemy of the original investigator who wanted to put them to shame and generate a result that was completely different. Actually, almost always, it was the same investigators who published the reanalysis as they had published the original paper. So what happened here? I guess that probably many more reanalyses were done. But in the current environment, the only chance, or by far the best chance to publish a reanalysis is if you say that I found something different. Even if you say that I found something different compared to what I found a few months ago, which is very confusing, this is really the current coinage, the current incentive in science. You need to find something different, even if you refute your own self. Containing the influence of conflicted stakeholders and authors is also a very big enterprise. As I mentioned upfront, the majority of papers in biomedical research still do not mention anything about conflicts of interest. This doesn't mean that there are conflicts of interest. I'm not thinking of some such conspiracy theory here, that all papers have conflicts of interest, although obviously all papers have some conflict of interest, including all of my papers, because I want to find something that maybe I have in mind, and I have a specific type of investigative plan that I have in mind. But I'm not talking about that. I'm talking about conflicts of interest that go more deeply, and for example, they could be financial conflicts that could have a major impact. And then the question becomes, who should be the sponsors and the authors of some type of sensitive studies, like randomized trials, meta-analysis, cost-effectiveness analysis, guidelines? New England Journal of Medicine, for 20 years now, has not been publishing cost-effectiveness analysis because they say they're so susceptible to the baseline assumptions and the modeling that you can get almost any result that you want, so I'm not going to publish any of them, which is, I think, a very radical approach because there's a very valuable design. Meta-analysis, we see lots of meta-analysis sponsored by the industry. Randomized trials, the default is that they're sponsored by the sponsor of the manufacturer of the intervention. So is that a good idea, or should we try to dissociate sponsors from the research that is being done? Better statistics and methods. I can spend hours on that. There's clearly tons of things that could be done. Many of these are field specific. But in general, I think that statistical training and improved literacy and numeracy of the scientific workforce could go a long way towards improving many of these problems that I showed you. Most of us, we are trained to keep up to date in a skill set that pertains to the subject matter that we're interested in. So we need to be acquainted with the new chip that appeared, with a new technology, with something that can help us measure 100 times more, 100 times faster, whatever we're measuring. And less so on keeping up with statistical methods, with study design, with caveats about statistical methods and study design. So most of the time, there's plenty of room to improve on study designs being used. Most of the time, there's plenty of room to introduce some standard features, like, for example, in animal experiments, randomization and blinding of investigators. They should be the default. There's no reason not to do that. And NIH and many other funders are increasingly experimenting with promoting the use of checklists, that for preclinical research, you need to fulfill a number of items so that we get a sense that what has been done is reliable. But the question is how exactly to do it. I think if we just throw checklists to people, people will have a normative response to that. They will say, okay, yeah, check, 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 check. Yeah, I did that, more or less. Will we really improve science? I think we need some studies to find out. So here's one example. We know for ages now that a randomized study should be randomized. And when it comes to animals, there's no reason not to randomize. And it's not that it's so difficult to do it with dice, with a computer, random number or pseudo-random number generator, whichever way. Nevertheless, even in that sample, even in the most recent studies of animals, only about 25% of them were randomized. Or we know for a long time that it's good to have blinded assessment of outcomes. So you want to see whether these mice are depressed. Can you judge mice that are depressed? You'd better be blind to not know whether that mouse was exposed to the intervention or not exposed to the intervention. Otherwise, I may get depressed, but I don't know about mice. So another major issue is that maybe we should be happy with small and tiny effects. If we push investigators to come up with big effects, maybe poor nature doesn't have that many big effects out there for us. Maybe it has tons of small and modest effects that are wonderful to discover, and they could also make a difference in our lives. If you have lots of them put together, you could save lives, you could improve lives, you could have faster technology and improve multiple outcomes. This is an empirical study where we looked at risk factors and interventions that have relative hazards less than 1.05. There are several of them, and some of them are truly genuine. So fruits and vegetables are a wonderful thing to eat more of, and I love them. But if you ask me what is the estimate for reducing mortality or actually reducing cancer risk with fruits and vegetables, The best studies suggest a hazard ratio of 0.999 per serving, which is 1 in 1,000. So if you tell me that I'm only going to fund you for finding hazard ratios of 0.5 or less, I would have to let go of fruits and vegetables that has a very nice 0.999. It translates to tens of thousands of people avoiding cancer and many more probably avoiding cardiovascular disease in a global level if I had one more serving per day. But it's not a 0.5, it's a 0.999. This is another empirical evaluation where we looked across the Cochrane database of 85,000 forest plots and 240,000 clinical trials worth of data, trying to ask how often we see very large effects in medicine. So if I were to ask how often do we see a 5-fold reduction in mortality seen in 2 studies with a p-value of less than 0.001 in randomized trials that have no clear evidence of bias, out of 85,000 meta-analyses that were seen in 1 case. And there's probably another dozen or 2 dozen that Paul Glasziou has tried to put together that nobody has tested with randomized trials because they were just so obvious. Well, we think that they were so obvious. So, you know, if I have a heart attack and I'm unconscious on the ground, you will do something. Yes? No? Okay. No, you can let me die. I'm such a miserable talk. But probably you will do something. You will resuscitate. There's no randomized trial that you should do that. But there's a few things like it. Now, there's many situations where we do come across big effects. We do come across odds ratios of 5 or more, actually 10% of the time in the randomized trials that we do. But practically always, almost always, when we run a much larger study, that goes away. Either completely or something much, much smaller remains, because these odds ratios of 5+ emerge out of studies that have on average 14 events. You know, it's just expected to be hugely inflated. Another major improvement is on the side of reporting research. So if you go to the Equator site that Doug Altman launched at Oxford a number of years ago, currently there's more than 200 standards of how to report research for different types of design. So there's CONCERT for randomized controlled trials, there's PRISMA for meta-analysis, there's START for diagnostic tests, there's STROVE for epidemiological research. And I think that this has been very helpful. These standards have not been adopted necessarily widely or equally across the literature. I think there's plenty of room for improvement. Not all of them are up to par in terms of how stringently they have been developed and how much evidence has been included in their background development. But I think it's a step forward, both for educational purposes and for transparency, for reviewers, for editors, for authors, for readers to know what is essential about a randomized trial that we need to know about if we want to understand what happened, whether we can trust the results, and whether we can take them forward, or for any other type of design. Many efforts are happening in the last few years trying to perform reproducibility checks. So Brian Nosek has launched the Center for Open Science. I showed you some data on the replication efforts and reproducibility efforts in psychology. He has also set a system where you can get badges for a study, a badge for sharing the data, a badge for pre-registering, for transparency, and for replication. The reproducibility initiative is another such effort where there's currently an ongoing effort to reproduce the most highly cited papers in cell biology and also highly cited papers in prostate translation research and other fields. There's multiple efforts around the world that are trying to test and probe reproducibility in very different fields. So about a year ago, we launched the Meta Research Innovation Center at Stanford, along with Steve Goodman and a number of other colleagues at Stanford, as an effort to try to become a connector hub of such initiatives, not only locally, but also around the world. It's very common for most of us not to know what is happening in other fields. And as I said, many of the challenges could be very similar. What is happening in psychology may be very relevant to what's happening in medicine. What is happening in astrophysics may be relevant to genomics and so forth. But obviously, most astrophysicists have no time to take a look at what's happening in genomics, and most geneticists have no time to look at what's happening in psychology. So there's a huge literature, an expanding literature, that is focusing on research about research. And just in the first 4 months of 2015, we came up with 815 papers in our searches, which I believe have only modest sensitivity. So maybe it's about 1,000 plus. And that means over 2,000 papers per year focusing on research with empirical data, with efforts to test some of these solutions and try to make progress. These are the 5 major themes that I believe are included in that meta-research area. Methods, performing research, reporting, communicating research, reproducibility, verifying research, evaluation, evaluating research, and incentives, or rewarding research. And I will spend the last few minutes on specifically rewarding research because I think that it could make a difference. So many scientists have argued, and that includes also a paper that I wrote with Moin Curia a number of years ago in Science, that if we want to improve validation, we need to reward validation. If replication and reproducibility is seen as me too and just waste, people are not going to do it. So we need to make sure that people understand that it is an integral part of the reliability, translational potential, and importance of the research enterprise. So maybe we need to think of how we re-engineer the reward system. And to do that, I have thought of the acronym PQRS State. It sounds like an electrocardiogram. So P is productivity, and productivity is nice. This is what people are rewarded for most of the time. So number of papers, but obviously currently there's so many journals that I can easily publish 1,000 papers by the end of the week. I just need a computer program to write these up. Fake papers and disseminate them to all these people who want to get $2,000 per paper published. But I need a little bit of money though, but yeah. That's the only caveat. So maybe productivity needs to be assessed in other ways. It needs to be seen, for example, if I do get funding, do I publish what I was funded for? If I register a protocol, if you ask me 2 years after I completed the study, did I publish that? It's a sense of productivity, but a little bit of a different twist. Or productivity, not just any paper, but papers that really had a major impact. So focus on the top 1%, 5%, 10% of even citation impact as a start for the specific field of interest. Q stands for quality. So each field has to agree what are some key quality elements that we want to promote. So animal research, we want to promote randomization and blinding of investigators, and a few other things. So judge based on these quality items. R stands for reproducibility. Proportion of publications that are reproducible. That could make a big difference. S, sharing of data and other resources. If I share, I get rewarded. If I don't share, I don't get any bonus. T, translational impact. Did that research get translated into something useful? Not for blue sky science, because obviously nobody has a clue what exactly would emerge and when it will translate to something useful. But lots of scientists, including myself, we promise that we will deliver something. So, I mean, did we deliver that if we do promise? And did that really help? Did it change people's lives, duration of lives, quality of lives, or other outcomes? This is a very naive schematic that I tried to come up to discuss some of these issues in my latest PLOS Medicine paper. I think of rewards as an exchange rate of currency. So let's say that a good publication in a high impact factor journal as a first or last author is 1 point. Nobody cares if it's replicated, if it's refuted, if it's translated, doesn't care. Nobody gives you any points if you share data. Nobody gives you any points if you contribute meaningfully to peer review. Nobody gives you any points if you educate others and train others. If you get a grant, that's 5 points, so you can trade one R01 for 5 papers. Then you can buy other wealth items. With 3 good points, you buy an assistant professor position with 10 points, an associate professor with 20 points, a tenure professor. If you have a PhD student, you get 2 points because you can sell your PhD student and get 2 papers. You can sell 2.5 PhD students and get an R01, roughly the same equivalent. And if you have power, you you're lobbying, you're president, dean, I don't know, you have lots of money, you can win up to 200 points by doing nothing. And then how are you going to change that? Well, one option is to give no value to publication, but give 2 points if the publication is replicated, give 5 points if it also gets translated, and lose 1 point if it gets refuted. 2 points for sharing, points for peer review, points for training, and no value for lobbying and for networking. And then there's the last column that is even more disruptive. In addition to what I described in the previous change, first of all, there's no value in titles. Everybody's equal. There's no professors, no students. Everybody's just scientists. And if you get grants, you lose points. So for each R01, you lose 5 points. If you get power, you also lose points. You can lose up to 200 points. And why is that? Well, if you have power, if you have money, you need to deliver. So then you don't want to get grants. You don't want to become a leader. There's a problem here. I mean, who's going to want to do research then, and who's going to want to become a leader? So I'm not saying that disruptive reengineering is necessary, but we have to think of what exact changes we need to make to try to arrive closer to the goal of getting more accurate, more reliable, and more translatable research. And in doing that, we have to think who else is in the game. So it's us scientists, but there's other entities and other stakeholders. There's the industry, which you can think of industry as sales of marketing, but also R&D. There's private investors, hedge funds, public funders, some of them open, others closed like the military, not-for-profit funders, philanthropists, journal editors, for-profit publishers, professional and scientific societies and universities, not-for-profit research institutions, supporting staff that want to make a living, hospitals that want to make more than just a living, insurance that want to make even more than just a living, and government, state, federal authorities, and people who want to live longer, they want to consume and to feel better. And all of these stakeholders have different priorities. Some of them want publishable research, others want fundable research, some want translatable research, and finally some just want profitable research. So we need to find ways to align the interests of all these people in science, and this is not very easy. To conclude, there's many possible interventions that can improve the efficiency of research practice and the reproducibility of the scientific literature. Few of them have strong empirical support. So much of what I showed you, I tried to use some empirical evidence, but I wish I had more to share with you. So be skeptical about what I told you. Well, I'm talking to the right audience, apparently. Empirical research and research would be useful not only to assess the prevalence of problems in research practices, but also to assess the effectiveness and potential harms of interventions that try to make research more reproducible. Because eventually, the adoption of optimal research practice can be key to the success rates, efficiency, credibility, and legacy of entire scientific fields. I'm closing with a slide with special thanks to some of my collaborators that participated in work that included empirical data that I showed you today. It wouldn't have happened without them. Thank you.
We have time for one question.
Only one question, that's a lot of pressure. I just had a question specifically about the end of your talk, which I thought was great, talking about how we changed the reward system and encouraged more scientists to do good science, basically. But my question is specifically about the T, the translational impact. I think we already have a bit of a problem with researchers making over-the-top claims like I'm going to cure cancer in order to pursue funding, and also a problem with the public and politicians not understanding the value of science that might be complicated and not have immediate impact on human activity. So how do we keep that, like, is it worth keeping that as a reward if it's going to have those negative impacts on the science?
Very nice comment. So I would not urge scientists to promise to cure cancer all the time, which we do promise all the time. I would only make sure that if someone finds something that cures cancer, they would get credit for that. So, I think that we do see some very major successes happening now and then, and we should go back and reward these people who follow these paths and led to these translational successes. I think it's a very bad idea, actually, to ask for every grant proposal to say that I will deliver something. I see that as leading to misleading promises much of the time, and it can cause lots of problems. So I would say it's perfectly fine for research not to say that I'm going to deliver anything. I'm just going to do my best. This is my ideas, this is what I'm thinking of doing. Or sometimes maybe even say I just want to explore, and I have a track record of knowing how to explore, and I'm well trained to do that. Go for it. If there is a deliverable, I think it's a different type of research. The promise should be very accurate. And I would argue that many of these types of research that have deliverables probably should not be funded by public funds. They should be funded by entrepreneurs, because they want to make sure that it does deliver. And then I would have public funds spent on the side of testing, very objectively, that promise that now people said, I have made it, and I have found some cure, some solution.
Do one more question. I can set up over here. Go ahead.
In your initial, one of your initial slides about all the pressures and all the sources of error, I noticed that two are missing, and I want to focus on one of them. One of them is confirmation bias. I talk to scientists all the time, and they only look for confirming evidence. They don't even think about looking for disconfirming evidence. So there's general confirmation bias, but more important, I want to ask a question about fraud. You haven't mentioned that at all. It isn't in your list of challenges. And I was on the investigation committee for a major university looking at scientific misconduct for a few years, and I was appalled to see how much fraud there was and falsifying of data that even came to light in that university, and maybe there was a lot more that didn't come to light. What role do you think that fraud and falsifying results plays in this whole picture?
Very nice question. So confirmation bias, I agree, is a very major player, and much of the action, the empirical action that I showed, probably has its roots at some sort of confirmation bias. The terminology can vary, so I like the term significance chasing bias. All of these terms are a little bit overlapping, but clearly, even with good intentions, I want to confirm what I have found, and there's some unavoidable bias there. Fraud does exist, but I don't think in the big scheme it's one of the major explanations for the problems that we face. Whenever we recognize it, it hits the news big time. It creates major reverberations because the trust of the public, of other scientists, of institutions is shaken. So in terms of the reverberations, I think it does have maximum impact. In terms of how much of the scientific literature is wrong because people were fraudulent, I think it's a very low percentage. If I were to put a number, I would say probably 1% or so, with some variability across different fields. I just don't want to believe that someone becomes a scientist, which means spending about 120 years in training and having miserable salaries and you know, being always under pressure, will I get my next grant to be able to survive, in order to become a fraudulent type of person. I mean, there's so many other things. You know, you can become a politician, or you know, and make tons of money. So I think that fraud does happen. It's something that we need to deal with, and we need to be alert and have processes in place. But the prevalence of, let's say, questionable research practices, which are not fraud, but they're cutting corners and doing things suboptimally, not the best way, I think are far, far more prevalent.
All right, that was the last question.